Deep within the microscopic world, a fascinating process unfolds. Imagine molecular machines, tiny but powerful, with the ability to cut the very essence of life, DNA. These are restriction enzymes, nature's very own scissors for manipulating the code of life. Just as a surgeon uses a scalpel with precision, these enzymes snip DNA at specific points, opening up a world of possibilities for scientists. Their discovery revolutionized genetic engineering, allowing us to study and manipulate DNA like never before. This process, called restriction digestion, is fundamental to understanding genes, diagnosing diseases, and even developing new treatments. Join us as we journey into the fascinating world of restriction digestion, exploring its mechanisms and its profound impact on science and medicine. Prepare to be amazed by the power of these molecular scissors. Imagine a long, tangled thread representing a DNA molecule. This thread holds the blueprint of life, but deciphering its secrets requires a way to cut it into manageable pieces. This is where restriction enzymes come in. These molecular scissors allow scientists to cut DNA at precise locations, creating fragments of specific sizes. These fragments can then be sorted and analyzed, revealing valuable information about the DNA sequence. Think of it like organizing a library. Instead of massive, unwieldy books, we have neatly categorized chapters. This is what restriction digestion allows us to do with DNA, transform a complex molecule into manageable pieces of genetic information. To understand how restriction enzymes work, picture DNA as a necklace made of beads. Each bead represents a nucleotide, the building block of DNA. These nucleotides are arranged in a specific sequence, like a code that determines our genetic makeup. Restriction enzymes are designed to recognize specific sequences of these beads along the DNA strand. Think of it like finding a unique combination of colors on our necklace. Once this specific sequence is identified, the enzyme binds to the DNA and makes a cut, much like snipping the necklace at that specific point. This precision cutting is crucial. It allows scientists to isolate specific genes or DNA segments of interest by choosing enzymes that cut at those points. The result is a collection of DNA fragments, each representing a specific portion of the original DNA molecule. The discovery of restriction enzymes was a remarkable example of finding extraordinary tools in unexpected places. These molecular scissors originate from bacteria, single-celled organisms often perceived as simple forms of life. In the microscopic battleground, bacteria face constant threats from viruses, their eternal enemies. Restriction enzymes are a key part of their defense system, acting as sentinels protecting the bacterial cell. When a virus attempts to inject its DNA into a bacterium, restriction enzymes recognize the foreign DNA and swiftly cut it into harmless pieces, effectively dismantling the viral invader. This ingenious defense mechanism is crucial for bacterial survival and has revolutionized our ability to manipulate DNA. Section 5. The right environment, temperature, and chemistry matter. Like any skilled worker, restriction enzymes need the right conditions to perform their best. Temperature and the chemical environment play crucial roles in their activity. Imagine these enzymes as delicate instruments sensitive to their surroundings. Too hot, and they can lose their shape and effectiveness. Too cold, and their activity slows down. Each enzyme has an optimal temperature range where it functions most efficiently, much like a Goldilocks zone for molecular scissors. The chemical environment, often maintained by buffer solutions, is equally important. These buffers ensure the correct pH level, providing a stable and optimal environment for the enzyme to work its magic. Without the right conditions, the enzymes might not cut DNA efficiently, or worse, they could cut in the wrong places, leading to unusable results. Section 6. Gathering the tools, our molecular toolkit. Before we embark on our DNA cutting mission, let's gather our tools, the essential components for a restriction digestion experiment. Our molecular toolkit includes 1. DNA sample. This is the genetic material we want to analyze, extracted from cells and purified. 2. Restriction enzymes. E. coli and HindT are two commonly used enzymes, each recognizing a unique DNA sequence. 3. Buffer solution. This provides the optimal pH and salt concentration for the enzyme to function. 4. Incubator, a controlled environment maintaining the ideal temperature for the enzymatic reaction. 5. Electrophoresis equipment. This separates the DNA fragments by size, allowing us to visualize the results. With our toolkit assembled, we are ready to delve into the intricate process of restriction digestion, a technique that has revolutionized molecular biology. Section 7, step-by-step performing the restriction digest. Our journey into DNA manipulation begins with the restriction digest. Let's break down the process. Step 1, preparing the reaction mixture. Combine DNA, enzyme, and buffer. This creates the perfect environment for the enzyme. Step 2, incubation time. Place the tube in an incubator. The enzyme binds to the DNA and makes cuts. Step 3, stopping the reaction. Add a solution or heat inactivate. This ensures defined DNA fragments. Step 4, visualizing the fragments. Use gel electrophoresis. Analyze the DNA results. Section 8, glowing fragments, visualizing the invisible. The magic of restriction digestion becomes apparent when we visualize the results. DNA fragments invisible to the naked eye fluoresce under UV light. This creates glowing bands, each representing DNA fragments of the same size. Smaller fragments travel farther, appearing at the bottom. Larger fragments remain closer to the top. This pattern acts as a fingerprint, revealing DNA sequence information. Distinct bands indicate a successful restriction digest. Smearing can indicate problems like incomplete digestion. Sharp bands reflect the precision of the restriction enzyme. Section 9, a revolutionary tool from genes to medicine. The precision of DNA analysis has revolutionized fields from research to medicine and forensics. Restriction digestion is a cornerstone of molecular biology, enabling groundbreaking discoveries. It allows gene cloning and the creation of recombinant DNA molecules. This enables large-scale production of valuable proteins like insulin. DNA fingerprinting in forensics relies on unique banding patterns from restriction digestion. Comparing these patterns identifies individuals and establishes biological relationships. The applications of restriction digestion are vast and continually expanding. This tool has opened up possibilities to understand and harness life's code.